Welcome everyone. I am Nishan Hajra, Assistant Professor of NS College of Pharmacy. Today I am going to discuss about the synthesis of picric acid from phenol. So we all know that the phenol, its name is 2,4-dinitrophenol. So phenol is a yellow crystalline compound basically used in various purposes. For example, it is used as an antiseptic agent or sometimes it used as a laboratory reagent for protein analysis or sometimes the detection of the alkaloids. So picric acid is a very useful compound, so we need to synthesize the picric acid. So picric acid basically synthesis from the phenol. We can see that the benzene ring, when the benzene ring contains the OH group, it's known as a phenol, and the picric acid it synthesis by the nitration of phenol. Why the nitration? Because in 2,4,6 position NO2 group is added and we create the picric acid. So while performing the synthesis procedure, we need various types of chemical requirement. For example, the main chemical we require, that is phenol. 2 gram is phenol is required, and another 2 types of chemical is required, that is concentrated sulfuric acid and concentrated nitric acid. So we discuss step by step how we can perform the synthesis of picric acid. So first step is we took the conical flux and add the phenol inside the conical flux and add the add the sulfuric acid. So why we need to add the sulfuric acid because at first we perform the sulfonation. For sulfonation we add the phenol and the sulfuric acid inside the conical flux and transfer into the water bath, and we need to maintain the temperature at least 80 degrees Celsius for 30 minutes. And after the sulfonation process, we further need to cool the sample because it is a exothermic reaction. That means while performing the reaction, heat will generate and it may cause the explosion. For that purpose, we need to perform the cooling procedure. After cooling, we add the nitric acid, and this process is called as the nitration. And in this process, we get the yellow crystalline stain of the picric acid. And after that, we further need to heat the sample by using the water bath, and also need to cooling the sample. After cooling, we finally get the yellow crystalline picric acid, but we need to filter out the solution. For filtration purpose, we use the filter paper, and at first we add the water into the sample and then perform the filtration process, and we collect the yellow crystalline stain. And we need to perform the recrystallization procedure for purity purpose. In recrystalline procedure, the yellow crystalline stain is further mixed with the phenol to get the final mixture of the yellow crystalline stain. Then we need to dry the yellow crystalline stain to get the picric acid. So as we earlier discussed that the picric acid is a very useful chemical. It basically used as an explosive, and in pharmacy it is used as an antiseptic agent, or sometimes it is used as a laboratory reagent. For example, it used as a protein analysis purpose or for the identification of alkaloid. But nowadays picric acid is not used mainly in pharmaceutical industry because the picric acid is toxic in nature, and it is very difficult to handle the picric acid while synthesis the picric acid, because we need to maintain some protocol or maintain, like we need to use the gloves. Okay, so picric acid nowadays not used as an antiseptic purpose, and further we will discuss the another topic from the same syllabus. Thank you everyone.